Hello student, so coming back to chapter number 2, that is sexual reproduction in flowering plant. Here in this video, we will study about about outbreeding devices, about pollen pistil interaction, and about artificial hybridization one by one. Okay. So first, let us discuss about outbreeding devices. So here to understand what is outbreeding devices, first let us check out the types of breeding. So there are two types of breeding. One is inbreeding and one is outbreeding. And if we say inbreeding, actually it is a crossing or breeding which is performed between closely related plant or between two similar plant. Outbreeding, actually it is the crossing or breeding which is performed between unrelated plant, okay, or between different plant. And if we correlate this breeding with pollination, then we can conclude that self-pollination will lead to inbreeding and cross-pollination will lead to outbreeding. But how? Okay, I will explain it. We know that during self-pollination, what happens? The pollen grain will land on the stigma of the same flower, okay, or different flower of the same plant. That means crossing will be performed between the same plant, okay. So self-pollination will lead to inbreeding. On the other side, in case of cross-pollination, the pollen grain from one plant will land on the stigma of another plant. That means the crossing is going to be performed. The breeding is going to be performed between different plants. So cross-pollination will lead to outbreeding, okay. But what is the main point here? The main point is that in case of plant, if continued inbreeding takes place, it can lead to a condition known as inbreeding depression. Here what happens? The fertility and productivity of the plant will decrease. Okay, and we also know that self-pollination will lead to inbreeding. That means if continuous self-pollination takes place, it can lead to what? Inbreeding depression. So this inbreeding depression is just like an unwanted character, okay, so it need to be prevented. And if you want to prevent this inbreeding depression, then we have to prevent self-pollination in plant, okay. And if self-pollination is prevented in plant, then we have to promote cross-pollination, okay. So here we are going to discuss the character which are present in plant which can discourage self-pollination and which can encourage cross-pollination. And we also know that cross-pollination also lead to what? Outbreeding, okay. That means we are going to discuss the character present in plant which can promote outbreeding. That's why the heading is what? Outbreeding devices, okay. So let us check out what are the characters which are present in plant which can discourage self-pollination and which can encourage cross-pollination. Come to point number 1. In some species or in some plant, what happen? These anther and stigma are placed at different position. So question is why they are placed at different position. So actually they are placed at different positions just to prevent this pollen grain to come in contact with the stigma of the same flower, okay, and thus self-pollination will be prevented. Now come to point number 2. In some plant, what happen? The maturity timing of this anther and stigma is totally different. Here what can happen? Anther can mature earlier than stigma, or stigma can mature earlier than anther, and thus self-pollination will be prevented. Now come to third point. In some plant, you will find that the flower are unisexual, okay, and if the flower are unisexual, then there is no chances of self-pollination, and thus self-pollination will be prevented. Now come to last point. In some plant, what happen? You will find cell incompatibility, a condition, okay, a condition known as what? Cell incompatibility. So what is cell incompatibility? I will explain it. Here what happen? The pollen grain of a particular flower may land on the stigma of the same plant, okay, but now this pollen grain will not be able to germinate on the stigma of the same flower because of genetic factor, okay, and this condition is known as what? Cell incompatibility, okay. So these are the 4 points which are present in plant to prevent self-pollination and to encourage what? Cross-pollination, or in simple word you can say that these are what? Outbreeding devices which are present. Next, come to pollen pistil interaction. Here we will discuss how the pollen tube are produced and how pollen tube enter into the ovule where female gamete egg is present, okay. So what happened? At the time of pollination, the pollen grain from same plant or different plant, from same species or different species can land on the stigma of this particular plant, okay. And this stigma has the ability to select the right type of pollen grain. That means they can check whether the pollen grain landing on the stigma is of right type or wrong type. And if the pollen grain is of right type, then this stigma or pistil will select the pollen grain and they will allow the pollen grain to germinate on the stigma. But if the pollen grain is of wrong type, then the stigma will reject the pollen. How? By preventing pollen germination. Okay. And pollen grain and this stigma, they communicate through chemical component that they release. After the selection of the right type of pollen grain, what happens? The pollen grain will start absorbing nutrient from the stigma, and thus an outgrowth will be formed through germ pore, and that outgrowth represent pollen tube. That is, after the selection of the right type of pollen grain will germinate and produce pollen tube through one of the germ pore, and this pollen tube will go through stigma, style, and it will reach the ovary. And after reaching the ovary, definitely the pollen tube will enter into the ovule through the micropylar end. And we know that towards the micropylar end, synergy is present where projection known as filiform apparatus are also present. So this filiform apparatus will guide the pollen tube into the synergy. Like this, the pollen tube will enter into the ovule. So this is how the pollen tube enter into the ovule. And all this event from where, from pollen landing on a stigma up to pollen tube entering into the ovule, represent pollen pistil interaction. So this is everything about pollen pistil interaction, and this interaction is easily represented by this simple diagram. Next, come to artificial hybridization. We already know that hybridization is the crossing between two genetically different parents to produce a hybrid plant. Okay, so for artificial hybridization, we have to select parents having desirable character, and we have to cross it, and we have to cross the two parents to produce hybrid plant. Okay, having desirable character. But for successful artificial hybridization, two things are required. Listen carefully. First one is we have to use only the desired pollen grain, and second one, the female parent must be protected from unwanted pollen grain. I will repeat it. For successful artificial hybridization, two things are required. First one is we have to use only the desired pollen grain, and second one is the female parent must be protected from where unwanted pollen grain. Why it need to be protected? Because we are going to use only the desired pollen grain. Okay. So, to prevent the female parents from unwanted pollen grain, two procedures are required, and these two procedures are emasculation and bagging. Okay, now come to emasculation. Emasculation, actually it is a process in which the anther are removed. Okay, it is a process of removal of anther, but this anther should be removed before it matures, that is before the anther produce the pollen grain. Okay, so that self pollination can be prevented. By doing this, the female parents will be protected from unwanted pollen grain. Okay, and this emasculation should be immediately followed by bagging. In bagging, what happens? The emasculated flower in which the stamen is cut out, okay, the emasculated flower will be immediately covered by paper, plastic, or polythene bag. Okay, by doing so, the female parents will be again protected from other unwanted pollen grains. Okay, so these are the two procedures which we have to perform in female parents to protect the female parents from where unwanted pollen grain. Okay, after this, coming to the process, when the stigma matures, okay, the cover bag can be opened, and this stigma can be dusted with the pollen grain which are collected from the desired male parents. Okay, and after this, the flower can be covered again. Okay, and when the fruit begins to develop, okay, this cover bag can be discarded. So this is the process of artificial hybridization. Now come to another important point. If the female parents is unisexual, then definitely there will be no stamen, and if there is no stamen, there is no need for emasculation. So if the female parents is unisexual, only bagging will be required. Okay, don't forget this point. That is, if the female parents is bisexual, both emasculation and bagging are necessary, and if the female parent is unisexual, only bagging is required. Okay, so these are some points related to artificial hybridization. And before we wind up, let me remind you what we have learned in this class. Today we have learned about outbreeding devices, about pollen pistil interaction, about artificial hybridization. Okay, and this brings me to the end of this class, and thank you very much for your attention.